Hello, this is Hector Giovanni Muñoz-Ayala, and this is my bacterium, Bdellovibrio bacteriovorus, and this is a presentation for principles of microbiology, or known as Bio 250. Bdellovibrio was discovered by Stope and Pezzet in 1962, in which the discovery was actually an accident, and the way they encountered the bacterium was when they were trying to isolate bacteriophages of phytopathogens from soil separation. And ever since, the bacterium has been studied and researched on, where the name of its genus, Delaware, is based on its lifestyle, and it comes from a Greek word vela, meaning leech, which I will discuss its lifestyle on the upcoming slides. The morphological characteristics of Delaware, that it's a gram-negative bacterium, and its shape is vibrio, and its reference in its genus name, meaning it's a comma. And the cells of the bacterium is pretty small by 0.2 to 0.5 by 0.5 to 2.5 microliters. So it's very small, and you can see the image right there as a perspective. And it has a monopolar membrane sheathed flagellum, which can actually reshape during prey invasion. The species part of the bacterium's name, bacteriovorus, the vorus part actually comes from Latin meaning eating, so it essentially means bacteria eating, and that's what they do. And again, if you look at the image, you can see the Dellavibrio invading Pseudomonas, which we'll see its attack phase. This is the life cycle of Delaware bacteriovorus, where it's basically a predatory bacterium that relies on its prey as the source of its life. It goes through the attack phase where it seeks its prey. Once it finds its prey, the bacterium uses the BD1075 protein on an enzyme to alter the shape within inside the periplasm of the prey, so it can digest its contents and it can multiply. And once it does that, it causes cell lysis, meaning the prey bacterium has been lysed, and it releases new Delaware cells. And it performs this predatory activity under aerobic and liquid conditions. The importance of Delaware bacteriovorus in the environment is how it provides protection in organisms, which is essential for their health. Like I mentioned before, Bdellovibrio has been isolated from the soil, and that has led to research in where the scientists discovered how the bacterium can be found in other places in the environment, like fresh water, brackish water, sewage, and seawater, implying that it has a role in those places along with other microbes. As you can see in the image on the slide, is a rhizosphere where it shows the biogeochemical process within the plant roots, like the chemical gradients where ions diffuse in certain directions for osmosis and cell signaling for many biochemical functions of the plant. And you can also see the bacterial association where there's bacterial communities in the roots, and even within those roots, Delaware bacteriovorus is a probiotic to the plant, meaning it can fight off phytopathogens and allow the plant to grow without being infected with disease, which is how and why the bacterium is important to the environment. Delaware bacteriovorus only targets gram-negative pathogenic bacteria like E. coli, and it's very dependent on the prey cells for its survival, and even biofilms of high prey density is definitively optimal for the growth of Delaware. Now, this bacteria is biotic, meaning has been found living in the gills and shells of crabs, oyster shells, zooplankton, and submerged plants, in which they have the same function as in the plants to fight off any pathogens and allow for the organism to grow and survive, and it's even part of their microflora. Delaware bacteriovorus has an importance to humans also due to being a future antibiotic slash probiotic. Over the years, there has been issues with certain pathogens becoming multi-drug resistant to antibiotics due to us overusing them, as seen in the image on the right side of the slide, where you can see on the right plate 3 antibiotic samples not being effective towards the bacteria that was swabbed all over, in which the World Health Organization declared this as one of the three most important public health threats. And these multi-drug resistant infections are mostly caused by gram-negative bacteria like Escherichia coli. Due to that, Bdellovibrio bacteriovorus has been considered to research by scientists, where they tested the strain 109J on in Platonic cultures, in which they found that the bacteria was able to lyse E. coli, which is its favorite prey, along with Pseudomonas. This demonstrated that the bacterium can be used as a biocontrol for human pathogens, in which, if you can see in the image below, you can see the multiple medical uses, like treating oral infections, and even in treating wounds and burns, and respiratory infections, and intestinal infections. The non-medical is just showing like it can be used or is actually found as part of the environments in those like in fishes, farm animals, and agriculture, which was mentioned in the last slide. Along with testing Delaware bacteriovorus and multi-resistant pathogens, there has been some research done to incorporate the use of the bacterium within the food industry. Specifically, scientists in Italy performed an experiment where they purchased chicken slices from the supermarket and they cut them into further pieces and then placed them inside glass jars to be autoclaved for 15 minutes. Then they were assigned to test and control, where all the samples were contaminated with diluted E. coli cells, and the test samples were contaminated with diluted Delaware cells. In addition, a diluted nutrient broth was prepared as a parallel analysis. After that, they were left for a few days, where the results showed that Delaware did lyse E. coli in the meat. In the table in the slide, it specifically shows that with graph A demonstrating that within the 6-hour mark, the predator cells increased, which is labeled as the square on graph A. The prey cells also decreased at the same time, which is marked with the circle on the graph. However, graph B, which is the diluted nutrient broth, it had a greater predatory activity even within the first hour, and it kept on increasing, which at the same time, the prey cells even decreased more than the chicken slices. It was due to how Delaware bacteriovorus performs its predatory activity better on liquid conditions, since the bacterium needs to swim to find its prey. That's what happened with the diluted nutrient broth, in which the chicken slices didn't have a lot of water since it dehydrated over time, thus leading to decreased predatory activity. Despite that, this shows that the bacterium is a great candidate for prolonging the safety of meat-based foods and can even minimize the usage of chemical additives in the future. It's just more experiments need to be performed, like placing a water film on the meat so the bacterium can swim and find its prey, since it does it better under a liquid condition. Everything that was discussed in the slides demonstrates why funding research behind Delaware bacteriovorus must continue. It's due to how the World Health Organization declared multi-drug resistant bacteria as one of the three most important public health threats, in which I mentioned how the bacterium can be an alternative to antibiotics, since the bacterium was tested on Platonic cultures where it was able to lyse a multi-resistant drug E. coli strain, which again demonstrated how it can be an alternative to antibiotics. Not only that, there's also the fact that it's able to control spoilage bacteria like E. coli and meat-based foods, which can prolong the food safety. But it's just more experiments need to be performed to ensure that we can actually use it. Not only that, the bacterium has been found to be beneficial to the environment since it can fight off pathogens that cause diseases in plants, and it's just overall a part of ecosystems. And these are the references I used for my slides. And this concludes everything. And I hope you have a nice day.